So we got about 40 folks here. I think if you want to just give a brief little intro, Courtney, and get things kicked off and then put up the slides, I think we can get cooking.
Absolutely. Welcome, everybody. Good afternoon. Welcome to the Clinical Trials Methodology course. This is the introduction to the 2022 course webinar series. My name is Courtney Miller. I'm the project manager for the course. I'm here with the course directors, Dr. Gutman, Dr. Coffey, Dr. Moyer, and Dr. Conwit. Before we start, please take note of the Q&A and the chat features down in the bottom bar. Please make use of that for any questions that you might have. And I can go ahead and pull up these slides now.
And I will add that while this is the first in our webinar series, our major focus is providing some information and a dialogue regarding our project-based course, for which there's competitive applications for, which you may have heard about. So I think while that's coming,
please let me know if you see the correct view.
I think if you click slideshow,
See the slideshow or the presenter view?
I see the presenter view.
Okay.
I think there's probably a way to switch it, right?
There is. How about now?
That's perfect. All right, so you can go to the next slide. We have our course directors here today. Other than Dr. Roger Lewis was unable to attend. He is professor of emergency medicine at Harbor UCLA. We revamped these slides, but we failed to adjust Dr. Lori Gutman, who's chair of neurology at Indiana University, no longer at the University of Iowa. My fault on that. Dr. Chris Coffey is at the University of Iowa, and Dr. Robin Conwit, who's program director at the Clinical Research Cluster at NINDS. All right, now you can go to the next slide. So what do we try to do in the Clinical Trials Methodology course? So first, we try to identify promising investigators in the clinical neurosciences and give them a rigorous foundation in how to design, fund, conduct, monitor, watch, and perform patient-oriented clinical research with a focus on clinical trials that deliver a intervention, some form of a healthcare treatment, as opposed to pure epidemiology. We do want to promote the ongoing professional career development by supporting the participants before, during, and after the program, so that they can continue to contribute to clinical trials to aid in discovery and help us learn how to take care of future patients better. We also have a goal of helping to design scientifically sound early phase clinical investigations that provide the foundation for pivotal confirmatory trials that can truly have an impact and reduce the burden of neurologic disease. You can go to the next one. So the course is designed to address a gap. Early trials are hard to do, and sometimes we do not dedicate as much planning and thinking to them as we we would like. We know that local mentors are busy. Resources sometimes are not evenly distributed across departments, medical schools, or the country in general, particularly in neurologic interventional trials. This can represent a key challenge to investigators working to get started in clinical trials. A key issue is that sometimes we get advice to do early clinical trials that aren't going to actually answer a scientifically interesting question, because we want each of these trials that is done in the early phase to not simply confirm our biases, but actually address uncertainty so that they can help us and other investigators figure out what to do next. Just because they're not practice changing doesn't mean that they're not important. And the idea is to put things onto a pathway towards doing practice changing trials and developing new treatments. Go to the next slide. So thinking of this from the perspective of the aspect of the course that is the project-based course, where you're, and we'll talk a little bit more about that in subsequent slides, where you as the investigator who is new to designing your own clinical trial is getting into the project-based part of the course where you're in a small group and there are mentors with you. These are some key expectations in terms of what sort of people would be the best candidates for the course. We're looking for people who are going to be the scientific leader of the project. So a couple very pragmatic ways of looking at that. Are you going to be the first author on the paper that comes out of this project? Yes, the answer should be yes. Are you going to be the PI on the IRB application? The answer should be yes. You should be at a stage in your career where you could potentially conduct a trial like this. So you should also have the tools locally, including a local mentor, to help design and improve and implement the study you propose. And again, with respect to a mentor, sometimes you may have you may be in a rare disease and you have a content mentor who's at another institution, and that can certainly work. But you need to have somebody who knows the ropes of doing clinical trials at your institution, because there's usually a lot of local context that can both facilitate and present a barrier to launching a clinical trial. You go to the next slide. So as I was starting to mention before, what we are seeking applications for is this blended experience where approximately 18 to 20 folks are going to get embedded into project-based small groups. There will be 3 or 4 trainees in each group, and each of these groups will be led by usually 2 clinicians and a biostatistician who has experience in the design of neurologic trials. And by design, your small group leaders may not be from your specific area of your specific subspecialty. This is not dissimilar from how a study section may react to, or maybe looking at a grant application or an FDA panel when reviewing a drug is not going to be entirely populated with subspecialists. You meet by video teleconference in the spring and fall. The spring is the key portion of starting to build a protocol. We do also hold large group public webinars. You may have attended some of them before, where we talk about general concepts in clinical trials, and to a certain degree, somewhat in grant writing, although clinical trials is our focus. We do hold some office hours where the course directors make themselves available for questions from the small group faculty and the participants, and even the participants' local mentors if desired. Then this year, we are back to having a residential course. It is going to be in Iowa City in July, and the dates will be there on our application. They will also be on the subsequent slides. At the residential course, you make a great deal of progress on that clinical trial idea, iterate it with many experts from across the country who are attending also in person, and we have specific didactic coursework with some of our other clinical trial professionals to help you really accelerate the progress on that protocol. Then we shift in the fall into a grant writing mode where you take your great idea, your great scientific protocol for a clinical trial, and turn it into a persuasive document to ask for resources, funds to do a study. This might be a grant that's going to NIH. This might be a grant that's going to your local CTSA, or this might be a grant that's going to a foundation. It might be part of a project for an AAN career development grant. We will work with you through those potential scenarios. Then for those who are able and are at the right place in their protocol and idea development, who've submitted a full grant, we will have a mock review panel where we do a simulated NIH style study section. In addition, for those who maybe were more at the point where they developed the protocol but reached some roadblocks in terms of getting to grant submission, we will be holding a mock IRB where the focus will be on some of the ethics of the trial, but some of the science as well. So we had that in the early years of the course. We're bringing that back this year so that many of you, so that a larger portion of you can participate in a summative experience at the end. And theoretically, if you wanted to participate, if you had a grant and you did want to put something into the mock IRB panel, if we had the space and investigators to staff that, that's also a possibility. But I think if it's, there is an ethical review as part of the mock study section panel as well. All right, you can go to the next one. So we've been very fortunate to be funded by NINDS. Initially, we were through an R25 mechanism, although this is dissimilar from the R25 mechanisms that some of your local institutions may have that are focused on a specific individual, usually in residency, to develop them in research. Our R25 was to distribute development across the country. The really great thing about this is it allowed us as the course directors some time for effort. It allowed us to compensate some of our faculty and allowed us to make the course cost-free to you as the selected trainees. So that was an investment by NINDS, and it's been an ongoing investment. The portion of our money to put on the course is part of Chris Coffey's Data Coordinating Center grant for the NeuroNext network. So in 2022, we are we'll be excited to be, bringing up our 8th overall cohort, and we are expecting about 20 trainees. There is some instruction regarding whether there's a biomarker track. This is something that we are open to. We've had folks who've submitted proposals that have a biomarker element. There's a lot of stuff on our FAQs about what types of biomarker studies are potentially appropriate. There has to be a very close linkage to a therapeutic or a therapeutic strategy. So if it's a biomarker that's purely prognostic, like if somebody has a bad value on this, they're going to do badly from their stroke, or we're going to look for things like that, that wouldn't be a good fit for the course. There may be good reasons to do such research, but our course is set up towards developing treatments. So again, if there's questions about that, we're very available and really would like to talk to people before they put in applications if they have questions about whether we think things would be a good fit, because we can usually help you understand that. And I guess I will just sort of make that point a second time. You could have a great scientific idea that is a bad fit for our course. And that's, we know that. And it's mainly because the structure of our course is really couched towards developing testable interventions and developing clinical trials to learn about what we should be doing next, both on a research perspective and for treatment. So that's a common theme. And again, if there are questions that emerge, please feel free to use the Q&A function. All right, you can go to the next slide. So what ends up happening? What's the level of commitment? There is a time commitment. We as the small group leaders expect some iterative progress on your protocols as you move through the course. There'll be small group meetings approximately every 2 weeks. We shoot to have about 8 to 10 of them in the spring prior to the residential course in July. And that there's a number, there's more than 16 weeks sort of in between. So it might be a little less than every 2 weeks, but generally it ends up being about every 2 weeks. And we don't want the meetings to be so quickly you don't have time to iterate your ideas, but we don't want them to be so far away that you lose momentum. So there are assignments in terms of your protocol development associated with these meetings. The open webinars that we talked about before, looking at more fundamental issues in clinical trials and clinical trial design, will be going on, and those will be recorded and can be looked at either synchronously or asynchronously. And then we hope that you have your protocol in complete draft form before the in-person course. The attendance at the in-person course, we use the terminology on the application, is expected. If you know for sure you're going to have a structural conflict, you have a wedding you're going to during that week, or you're going to have to be covering a clinical service, this wouldn't be a good year to apply for the course. Now, we know that people may fall into emergencies or need to quarantine or things like that, but the expectation is that all things being equal, if you're accepted into the course, you're blocking out those days and you're going to be there in person, barring unforeseen circumstances. We then kind of go on pause through the rest of the summer usually, and then start to meet again in September to take what has been this great protocol you will have developed and then start to translate it into the form of a grant so that it can be used to get you the resources to actually enact that study. We do expect that the course will shape and change your idea. So if there's a lot of certainty, it's a very late stage idea, it's been well vetted by you and your mentor, and there's not a lot of flexibility to change, the course might not be a good fit for you. So we want to make sure that you're potentially have the ability to do things. It's not to say that you have to always take our advice, but sometimes we identify issues that will strengthen the trial design, and we want to make sure that you can make progress and learn from what you're learning in the course. You can go to the next slide. So we have a little bit here on some of the outcomes. Now that we've been doing this a few years, we're still trying to track down a few of our trainees from over the years. And we have additional data on how long it gets them to get to some of these outcomes. But right now, based on everybody we're still continuing to follow, about 2/3 of the trainees either got or are working on IRB approval for their CTMC project. And that, I think, is a good number. I wouldn't be daunted by that number. Sometimes one of the most important things of a trial planning exercise is an understanding that you can't do the trial. You need to do a different type of study or a different trial. And the fact that we get some investigators there is, I think, good for them and good for science. If they might have embarked on a study that would have had feasibility issues or may have led to ambiguous results that wouldn't have moved the field forward, it's okay. Although when we see that things are going in that direction, we do hope to help you pivot to another idea. But this has been our experience. So I think this is a good amount of success in this. We know that across academic health centers, many investigator-initiated studies, don't get to the IRB approval step, don't get patients enrolled. So I think we are able to add value for you in terms of increasing the likelihood that you're going to get your study going. You can go to the next slide. Of the people who have participated in our course in this iteration of the training in the general development of an early phase trial, about 57% of them have submitted an NIH grant as either PI or multiple PI. And this would include K awards or awards of any type. You go to the next slide. And amongst those 57% who've submitted a grant, which, as I had been told by a admired project director at NINDS, John Marler, you can only get a grant if you apply. Amongst those who had submitted a grant to the NIH as a PI, about 55% of them have gotten a PI grant. And only 10% were no longer trying at the time that we had checked in with them. So people are working on it, and I think that's something that, again, I think clinical trials can be challenging. They take team efforts and oftentimes a long scale in terms of your career development. So, this is something that I think I am proud of as one of the course directors in terms of getting people qualified to submit NIH grants. You can go to the next slide. So the application, we have frequently asked questions, and again, we will certainly be willing to talk to people or answer emails. The one thing that I will say is once, the due date's February 28th, my ability to answer emails or talk will, the amount of availability I will have is decent right now, but on February 25th, there's going to be less. So that as you look through the application, anything you can do to do it earlier, and if there are things that you want us to look at, we will do our best to spend some time to give you some feedback on things. But the most, the things that we really want to know about is what's the area that you want to study. And one of our most important grading criteria is that it's delivering an intervention, a drug, a device, a diagnostic, a behavioral intervention to patients in some prospective way. If that's not part of your application, we typically are not going to score it very highly, which is why if you have questions about scoping, getting those out of the way early is really important, because we would rather not have to have you be sad that we couldn't take you in the course, and us be sad that you submitted something that we sort of asked, told you that was a little bit away from the goals of the course. And again, I'll say it a third time, it could be a great scientific idea. It's just not the way that our course is set up to develop things. So thinking about the intervention, it has to have some form of a good basis in biology, or if it's a behavioral intervention, theory, or if the biology is less well known, basic epidemiology and other prospective studies. So the best designs for the course will seek to confirm important preclinical or theoretical estimations of dose, mechanism, or target acquisition. And again, that sounds very drug-y, but this could apply to behavioral interventions as well, in terms of how often are you going to text people to try to get their blood pressure down. So the goal is really to learn whether and how a follow-up trial should be conducted, and seeing that you're on a pathway to that with your current trial is really what we're looking for. We want you to think about the NINDS transparency and reporting guidelines. There's a link to that in our application. And this is really to think about, is the evidence that you're pointing to from preclinical experiments, was there a good amount of scientific rigor that got to this point before? And if there wasn't, are you cognizant of that and seeking to rigorously develop and develop new data that fills those gaps? The sort of this other part of the scientific area. So I get it; it's a lot of stuff to put on one page, but we appreciate having this in a succinct way. The critical summary of like what's the impact of the new thing that you want to do, and that's important. Again, that's a lot to get on a page, but we've had some good success in terms of finding projects, I think, that are very interesting using that methodology. You can go to the next slide. So then we have a bullet pointed summary of your research question. Sometimes, even with the best intentions, the first page becomes a little harder for us to understand. And this is where we just sort of drill down into bullet points, like, what's the disease? TBI. What's the phenotype? GCS 3 to 8. So this is really a way to even further clarify your ideas, so that when we're quickly looking through what exactly is it that they're proposing to do, we get it. And what's the main goal? What are you trying to learn? And what's the measurement that you're going to use? I'm going to look at brain edema in this TBI study. And we have an example that's filled out in the application, and some guesstimate of sample size. We understand that you may not have access to resources, but if you think, I think I'm going to need to do a 10-patient study, a 50-patient study, a 200-patient study, or some range, getting an idea of the scale. Typically in our course, if you want to do a 10,000 patient study, since we're more focused on early phase designs, that's typically not an early phase study. And it's typically not somebody who's doing their first, designing their first clinical trial. There are other resources through StrokeNet, NeuroNext, and for neurologic emergencies, the Siren Network, to develop protocols in those spaces. We do want you to also be aspirational in these bullet points as to, like, if you find what you think you're going to find, what do you do next? It's like, I'm going to confirm the dose, confirm that there's effect, And then I'm going to be doing a larger phase 2 study where I'm looking to see if a phase 3 study is justified, whether it's that's sort of what you're looking for, what the next study would look like. And and then looking even more aspirationally, how would the next study change practice? Would we get a new treatment out of this? Would we know what to do about future patients more than what we know today? And again, if you're proposing something that involves a biomarker, how the biomarker interacts with the treatments and not a formal statistical interaction, it's just like, how would you use it would you use it to guide treatment? Would you use it to tailor treatment? And how would that, or would it inform the clinical trial design by sort of confirming a biologic mechanism? You can go to the next slide. We want you to think about potential funding sources. And again, this is important. We want to have like sort of 3 areas. We don't want the course to be a pure exercise in developing a theoretical idea. We want it to be something that you can use, try to get the resources through grants or local funding and do. So we do look at this pretty closely. If there isn't a thoughtful way as to how you're going to get the resources to do the project, we do, that is one of our scoring criteria. So it can also help to go look at Project Reporter on it, through NIH to look to see if there's past grants that have been funded in this disease area, or if there's other people who are already doing the project you want to do, which may be a good sign that you may need to adjust your project if there is already a funded project in the space. But it gives a sense as to what the funding priorities are. I don't know if I have it on the next slide, but I will make the notation that each NIH institute is a little different. But at NINDS, some of the earlier, the smaller mechanisms like the R21 and the R03 are not, it's been found that you really can't do clinical trials, even small ones, with that amount of resource. So at NINDS, it's not permissible, or it's either not permissible or strongly discouraged to submit clinical trials into those mechanisms. The sort of first one for a project grant would be the R01, or other special, there's some other special larger scale grants. K grants that involve a clinical trial, the clinical trial has to have mechanistic aspects at NINDS. Now, that being said, since I'm in the research network Siren that also is neurologic, plus heart, lung, and blood emergencies, other NIH institutes that might be doing neuroscience work, the National Institute of Aging, the National Institute of Mental Health, they have different rules as to which mechanisms can be used for what sorts of things. So I would say that you want to be thoughtful here, but again, if it says, I'm going to submit an R21 to NINDS to do this study, we may have some feedback for you on that. That's probably not going to be a tenable plan. You can go to the next slide. Team members, these are the people, whether your mentor, coordinator, if you have data management resources or engineers, you can invite them to your small group meetings and how you will organize and interact with the team. We want biosketches, a sketch from you. We want to make sure that your personal statement addresses your motivation for taking the course. And your mentor's biosketch, there's not an additional letter from your mentor. So in their biosketch, they should address how they were going to mentor you and help you implement the project. You can go to the next slide. And you can have more than one mentor letter or mentor biosketch if you do have a team, like a content expert from afar and a local expert who can help with clinical trials. You need a letter from a chair or division chief, that's someone who has the authority to talk about your career development and your potential for a successful clinical research career, but also the institutional commitment to you. resources are there to help you implement a clinical trial protocol. Finally, if you have your heart set on, doing a study on an investigational compound, something that's not available commercially, but is in development. Maybe you're doing a stroke study and you want to use an MS drug that's in development as an example. You have to have some form of written commitment from whoever owns that molecule that they're really going to give it to you. Because we find that there are potentially a lot of barriers to that, from the risks to the owner of that molecule, of something going wrong in this other investigator-initiated study that's running alongside their study where they're trying to get approval for their drug, or other reasons. So just for that, even if you think you can get something, or somebody from industry or pharma has told you they can get you something, some form of a letter that would indicate that if you get your study, if you get your study designed, that you could actually get access to the thing that you want to deliver. And additionally, that it exists on Earth. Sometimes people have an aspiration, like it would be a little ambitious to say you're going to develop a new oligosense, nuclear anti, like if you're going to develop a new compound during your CTMC project, we're going to probably be like, so it has to be something that currently exists, as well. You can go on. So we've covered some of these things already. What are the key pitfalls? We've said it a few times. If what you're proposing isn't really a clinical trial, that's a problem. Now, there may be perspective. Now, just because you're not randomizing to two groups doesn't mean it's not a clinical trial. There may be historical data. You're trying to estimate a rate of complications. You're trying to estimate how much adherence there is to a regimen. So some things that might not meet the full NIH definition, although the NIH definition really is the prospective assignment of treatments. For something where maybe you're doing a single arm study to look at adherence, which might be a perfectly good design, that might not quite meet the definition because there really is an assignment. But from our perspective, if that study is well justified, that might be a good fit for the course. Like I talked about before, proposing a phase 3 study is a problem, just because that we feel that there are other mechanisms that are better venues for developing that. And if you're a good fit for our course, but you're also thinking about doing a phase 3 trial through maybe a multiple PI team, it's a challenge, because you're sort of picking, should you develop something a little bit smaller scale for this course, or should you go in on that project? And again, we can help advise you on those things, but within this project, we've not found that developing phase 3 trial ideas is productive, because usually we don't want to clash with the protocol development teams that would help you from StrokeNet, NeuroNext, or Siren. We'd rather get you into those folks as soon as possible, because they have a process to help you design those sorts of phase 3 trials. If there's not a really clear question, you put a lot of background, like this is a really bad disease and we're going to find something to do. That can be a little bit hard for us in terms of the application. So we do need some background. We want to know a bit, but we want you to also spend some time on what it is you're going to do. And again, even though I say lacking, putting too much background can be a problem. Having some data that's coming from rigorous preclinical studies or theory, there needs to be some reference to that. So one pitfall is a sole emphasis on safety. And again, you may have gotten advice that when you're doing an early phase trial, the most important thing to do is to show that this new treatment is safe. it presents a little bit of a lost opportunity because there are likely other questions at that stage that might be more interesting, whether it's feasibility, proof of concept, or effect. For example, I will use the example of thrombolytic trials in stroke. There's a pretty well-known approximately 6% hemorrhage rate for most thrombolytic treatment trials in stroke who are enrolling patients of the range of severities currently eligible for that treatment by guidelines. Doing a study where you look at 10 patients and you confirm that there was only one hemorrhage is not moving the scientific, of a new agent, is not really moving the scientific field forward much. You've sort of gone to a lot of effort to enroll those 10 patients and kind of confirmed that if you see 0 or 1 or maybe even 2, you're sort of within the confidence interval for 6 for a sample size of 10. So you've had a lost opportunity to actually answer a question that would be more helpful in terms of giving you the right tools to decide whether to do a follow-up study of that agent, but also funders and people reviewing your grants later as to why to do it. So again, a sole emphasis on safety might be appropriate, but it's usually not, because there usually are other questions about what you're using. And sometimes we see studies where it's a drug that's used a ton in some other condition, like it's been used in millions of patients, but then it's going to be adjusted for Parkinson's disease. And sort of showing that a drug that's been used in 1 million patients already is safe in a specific disease is also a lost opportunity. There's certainly better questions you can be answering there, unless there's some really compelling biological reason to think that this is a drug that's going to induce pathologic gambling, and it's been known to do that, and we're worried it's going to do it a lot more in Parkinson's. So there may be safety endpoints that are important, but you just have to, we really worry about a study where we can look at it and be like, yeah, I know they'll meet that bar. The study, they'll show that this is safe, but then what do they do next? So again, we can certainly, if that's the head scratcher for you, that's something to go. Oh, good question in the chat. Can 2 people apply from the same institute, or does it have to be 1 person per institute? We have no limit. We wouldn't use, while we would be sad if our entire cohort was 20 people from one institution, and I'm thinking that's unlikely to happen, we don't throttle applications in any way. We don't stratify. There's no limitations on people from the same institution, because people might apply from different departments. A neurosurgeon might apply, an emergency medicine doc might apply, a stroke doctor within neurology might apply, but a pediatric neurologist might apply. So, yeah, people from the same institution would certainly are encouraged to apply. And we've frequently at least had a couple people from the same institution in most of our cohorts. Yeah, so the next question is, could you describe your applicants, level of trainings, residents, mid-career faculty, fellows, specialty, academic background, et cetera? It's a good question. And I think in terms of the distribution of specialties and subspecialties, we probably, in terms of career stage, most of our applicants and successful trainees are in the assistant professor step, but I would consider early career. Now, sometimes we've had later career people or mid-career, or even a couple senior people who had a career that was entirely focused on say, basic science research, and now they're branching out into clinical trials. If there's a plausible case to be made that this is a new thing, learning how to do clinical trials is a new thing for you, we're open to the application. But from a equity perspective, a full professor who's done bench work for 20 years and is in a pretty good space professionally, we will, it would have to be a really good application, because we do also have a mission to kind of move up people in the early phase of their career who want to include clinical trials as part of things. So I would say about 55% of folks come through from neurology. The other 45% is a mixture of emergency medicine, neurosurgery, sometimes general internal medicine, people you're doing stroke prevention, PM&R, psychiatry, psychology, and and I guess probably even like a bit of a higher proportion, I guess, by saying neurology is about 55%. Child neurology would usually be like another 10 to 15% each year, although it depends. Some years are bigger than others. And then with the remainder through the other specialties that I mentioned. Neurocritical care is something that is also valued. And sometimes those folks, even though they're neurologists first, they're cited in departments of neurosurgery. So we thinking about people's departments. We've had trauma. That's right. We've had trauma surgeons. We've had radiologists, both on the diagnostic side and the interventional side. We've had pharmacists. So we've had a lot of. We certainly, if you're potentially able to sit as a PI on a clinical trial, and it's going to be in the clinical neurosciences, you're potentially eligible. Yep, we've had psychologists and both neuropsychologists and psychologists focused on trauma for families after TBI and so forth. So not to make sure that I completely answer Dr. Rafiq's question. We've sometimes taken residents, we've sometimes taken fellows, depending on the stage. One of the issues is just the amount, the focus in residency is on learning clinical skills, and usually in fellowship it is. There are often research years, sometimes depending on how it's set up, but it's been a little bit hard for residents to get enough of a running start and enough resources. There's certainly unique situations. Sometimes somebody's about to enter, there's a guarantee to be entering facultydom at the end of residency or fellowship. So the degree to which it's clear that the institution has a long-term plan for you if you are a trainee can help, but we've found that mostly folks in the pre-faculty stage don't really control their own destiny enough to fully benefit from the course, but everybody's mileage can vary. But we have a pretty good signal that the folks who've come as residents or fellows, their sort of success rate of getting their project to IRB approval, their success rate of getting NIH grants is a bit lower, although that may just be a time scale problem. We've had psychiatrists too. All right, those are all good points. I think so a pitfall is I'm going to start the course in April and I'm moving in July. Sometimes I will speak from experience in terms of moving right after residency and moving to a new city and having a child. I underestimated the degree to which my life was turned upside down by moving. And we think you probably are too. So if there is a move that is planned within the scope of the course, like you're going to move in July, then come to the course, do some planning in advance, probably not a good fit this year. I mean, we can talk to you about your individual situations. Bear in mind that the course directors, we almost sort of have like, it's almost like a little bit of a joke. Like, I'll be like, yeah, we can, I don't know if it's a great idea, but we can see what will work. And then Lori and Chris, who are, and Lori, who's now a department chair and has seen a lot more people do things over the years. Anyway, she would give you a slightly more direct answer of, it's a really bad idea. So if you're planning to move, I'll just give Lori's answer is it's probably a bad idea to do this course this year. You'll get more out of it if you wait. And the course is going to be around next year. So I would recommend that if you truly have a move planned, adding this to your plate, again, you can talk to us about why your case is different, but and I'll be sympathetic, and everybody else will be like, it's a bad idea. So anyway, yeah, and I think the other thing is sometimes we do get applications where there's an ongoing study. I said we have to have the flexibility to help you change your idea. If your study is already going, that's pretty hard to change. So again, that's another pitfall. All right, you want to go to the next slide? All right, here's the real nitty gritty. 28th of February, applications due. After you apply, we'll be asking you some additional questions. We are planning to be able to get through the applications by March 15th. If you get, we appreciate early applications. We're always a little nervous about accepting people early because we just don't know how many other things are going to come in. And we really take a lot of time and care to try to select the most meritorious applications that have projects that we think the course can help move along. Then after we've made these decisions, we're going to give you the ability to match into the small groups. The faculty will have, while you are writing your application, the core faculty are setting up times that they are going to meet, and you'll be, there'll be like the 5 small groups to choose from, or 6, and you will be like, rank these 1 to 6 based on the order, based on who's in the group, but also the time of the group, and you'll want to pick a group that you can attend. We've changed this from past years where the groups were set and then they had to negotiate a time to meet afterwards. And again, if you can't find a match amongst the times that we have, the 6 time blocks that our small groups will be happening, and we try to be sensitive to have some that are later in the day for people from the West Coast, then we may have to defer your participation to 2023. If you're accepted and can't match, we won't make you reapply, but we will make you, we do want to make sure that you have a time that you can attend. We'll start having the small groups usually in April, but possibly in May, depending on when the groups are available. We'll start to block out we're going to block out those times on faculty calendars in advance. And then once you get matched into the small group, you will be getting the schedule requests. We have a reunion at the American Academy of Neurology annual meeting. For those of you who are new to the course, obviously it won't be a reunion, but a potential chance to meet us in person, if you are there. And then the in-person course in Iowa City in July. All right, go to the next one. So this is just, ooh, not Ann Arbor. I missed something. We get it. We do it every year. We miss a couple things. I think I missed Lori's move to Indiana like twice. It used to be in Ann Arbor. It was in Ann Arbor a while ago. So this is, again, this slide is wrong. We can annotate this, but yeah, July 18th, 21. Arriving, so it's a Monday. You won't need to be there Sunday night or anything. You'll arrive on Monday and there'll be evening activities. There's, and again, sort of the small groups, August, we tried to do some August meetings last year. We can probably say that the proposal development, or the proposal development's up at the top, and the protocol development's up at the top, the proposal development's at the bottom. I think I got those a little tweaked out. But September through November are when those other small groups and mock study sections will happen. Before the pandemic, we were alternating between Iowa City and Ann Arbor, the major heads of the Clinical Coordinating Center for NET, the predecessor to the Siren Network, and the Data Coordinating Center for the NeuroNext network.
Well, there's a question about K awardees and when they should apply.
Yeah.
It went away just now.
Sorry, I just answered it. It's the question was, have you had K awardees in the past? If so, is there a recommended time to apply, year 3 or 4? And I just said that we have had K awardees, but no particular time to apply, as it depends on your K timeline, what you need to do, what kind of pre-work you need to do before you start your clinical design. So it really depends on your K of project. You have to figure out how it's going to fit in best. but you can certainly write in your K application that that one of your goals is to be a part of the course to design your study, to further design your study or refine your protocol.
Yep. No, that's a good point. All right, I think go to the next one. All right, I think people have been using the chat function, which is good. This is one of our large get-togethers in Ann Arbor, and those are our websites where you can find the application and other materials. Neurotrials.training is just a sort of alias that points to the Siren Network link there. All right, have you had or will you be open to accepting applications testing novel clinical informatics machine learning tools for diagnostication? I think you should probably I give a maybe. I mean, I guess diagnostication, I'm not sure what we want there to be.
I mean, address it because this ties to some other things, Will. I can try. I mean, it kind of depends on if it's far enough along where you're testing the efficacy of it, or if it's to the point where you're still trying to tweak it or figure out how to do it, then that's not a good fit. I mean, that goes for a lot of different things. It's kind of the equivalent of the dose for a drug. If you kind of know what you're doing and you want to do proof of concept of a specific diagnostic measure, it could fit. If you're still figuring out how to make the measure work, it would not fit.
Yeah, I mean, oh, go ahead, Lori.
I was gonna say, one of the things that you can do is if you have a question about it, is send us a short version ahead of time, and we can even have a discussion with you to see how it might fit into the course. So we're open to that. I think you mentioned that earlier, Will, right?
Yeah. So I guess
Thank you. I think can you hear me?
Yes.
Yeah. So I'm happy to clarify a little bit now. So yeah, it's gonna be so I'm developing it right now in phase 1 as part of a DoD funded project. But the idea will be subsequent phase 2 validation of this against the clinician assessed diagnostication in a multi-center study. So it's like a biomarker in some way. There is a prognostication aspect also to this, but the focus will be first for the diagnostication part. So classifying the patient on the formal diagnostic criteria of clinically probable versus possible through the tool versus the clinician.
So I guess the question I would have is what disease, and then what does that?
Parkinson's, sorry, yeah, I can mention that. Parkinson's disease. So there are MDS-PD diagnostic criterion, which incorporate ton of information, clinical imaging, exclusion criteria, inclusion. It's so complex. So the idea is to develop a more standardized, on automated way, the patient data is being captured through the system, and the system is incorporating it to coming up with that classification probability.
I see. So the idea would be these are people who don't know they have Parkinson's. You mine you mine their data and say, congratulations, you probably have Parkinson's. Is that
new patient who comes with, whom we diagnose based on our clinical examination, the same information is being captured through the system by the clinician, but the system is automatically parsing out that information through the algorithm it has, and then classifying the patient on the criterion in an automated manner. And the project will be the validation of that, in a multi-center study where you have blinded investigator diagnosing the patient using the approach manually versus the system doing it.
Yeah, I mean, I guess not to be a Debbie Downer. If I was thinking of them, I'll just sort of say thinking about the general concept of a machine learning idea, if it was, if I was to do a machine learning idea that I thought would be a good fit for the course, it would be maybe identify people who are in general care, but probably have medication overuse headache, and then send them Topamax. Again, that's a pretty again, I don't know if that's a really good idea, but at least there's a sort of clear linkage to one potential treatment for medication overuse headache is Topamax and other social things. Whereas it seems like the main endpoint in yours is helping to facilitate the diagnosis of Parkinson's, which does have some therapeutic links, but typically something that's completely focused on diagnosis is less of a good fit for our course, if that makes sense.
Yeah, I thought it would be like from a biomarker standpoint, because the RFA says you do entertain biomarker projects, but it seems like therapeutic trials are a priority from what it sounds like to me.
Yeah, exactly. Like, so I would say if the biomarker was to find people who had a specific subtype of Parkinson's that would respond really well to a particular treatment regimen, that's the sort of and would sort of randomize patients to actually be given a change in their therapeutic pathway based on that subtyping. That would be more, I think, in line with what the course would be designing.
Okay, thank you.
It's a good question. And it's a good like sort of illustrative example. Like the simple sort of examples are like, we want to, if it's a specific type of, kind of develop a biomarker that says I need to give more or less thrombolytic. I can tune the dose of TPA to be just right based on a panel of biomarkers, not give too much, not give too little, so that it's enough to break open the clot, but not so much to bust past the blood-brain barrier and cause a lot of naughty things to happen back there. All right, well, I think if there are other questions, certainly there's a general email on the application, and also my email, wmeurer@umich.edu. We are really excited to start to see your applications, and I've probably gotten some questions already that I haven't responded to, and I'm sorry if I haven't responded to any of your questions that you've emailed me already, but we will be better about looking through those things. And again, thank you everybody for being here. Thank you, Drs. Conwitz, Coffey, and Gutman. I talked a lot, so I'm glad that you all were here too, though.
Yeah, and this will be up on the website, recorded on the website if you want to go through the slides again. Thanks.
All right. Thank you, Courtney.
Thank you. And as Lori said, this will be posted on the Siren CTMC website by early next week. So thank you all. Have a great day.